So in iGEM, we have to somehow
I do know. Yeah.
disseminate our project. So one of way is to disseminate our project by having a discussion session with international experts, and we believe that your expertise would be a valuable insight to the team in our project. So can we ask more, because this we are made up of past biology Olympiad winners in Indonesia, and we established this organization mainly not for iGEM but for Olympiad training for further future competitors, and the winners will be eligible to undergo a selection process for an international scholarship, probably to study at Monash. And we think if you can share a bit about your research expertise, your background and interest, probably people who are interested in similar areas who actually get a scholarship to study here might want to talk or be part of your lab or your colleagues.
Yeah, sure. Yeah, so my big interest here, so I'm in chemical and biological engineering, and so we focus on pretty much everything from right down looking at the organism itself right through to how you might work with it in a bioprocess. We look at the sort of products you might be able to make, how to metabolically engineer organisms, not so much on the fine end of genetic modification, so we don't really develop new tools from that point of view, but we heavily use omics here where it's appropriate to help us inform us on the metabolic engineering success and where our blockages might be and where, all that kind of, those sort of problems. Primarily, I work with yeast, but we've also had some projects in bacteria. So, and those yeasts are, we use Saccharomyces heavily as our model, and then sometimes into Pichia, pastoris, or Eurovia lipolytica, mostly for lipid work. They are less understood in many ways, and there's less tools for them. So that's why we still have a heavy foot in the Saccharomyces camp.
Nice.
Yeah. So yeah.
Why do you choose to primarily work in yeast, not like, for example, E. coli? E. coli is like one of the best established model organism. Why yeast?
Eukaryotes offer opportunities that you don't get with E. coli. If you want to just make some bulk protein, we use E. coli. You burst it. If you can do an easy purification, no problem to get the protein. Most of the time, if you're wanting a very functional protein, maybe a higher value protein, it's usually a secreted protein and you need a eukaryotic organism, and particularly one that can do post-translational modifications.
Makes sense, yeah.
So, but that's not always the reason. But I guess if you think en masse around the world, probably the most used organism is Saccharomyces cerevisiae.
Right, I see.
Okay, so if you think about every winery, every brewery, every bakery, every bioethanol, drop of bioethanol made, that's all Saccharomyces. So it's actually probably the dominant biotechnology organism. Even though E. coli is fast and robust and excellent for a whole lot of things. Yeah, but I think Saccharomyces rules actually.
In terms of eukaryotic organisms, because some features of the cellular machinery doesn't work in prokaryotes, right?
That's right. Yes. Yeah.
And what kind of real world problem do you are you primarily interested in tackling with engineering these organisms?
So that real world problem might be something like making proteins. So therapeutic proteins, functional proteins. We don't work so much with therapeutic proteins, but certainly functional proteins like protective proteins that are in animal products, which we can now make via this vegan route using yeast. It sounds easy, but it's actually a little harder in practice to get high titers and to get predictable and consistent production. And so, so it sounds easy, but actually the real world part of that is trying to get economic production.
If you don't mind, I would like to showcase our design to you.
Sure.
So in this competition, we are designing a particular genetically engineered microorganism for tackling real world problems, particularly in Indonesia. And let me just open the presentation if you don't mind.
Go for it.
So basically, because we're going to use one of your model organisms, Pichia pastris. We're going to tackle two problems at once. So this is really an ambitious project. Hopefully it goes well. But what happened is,
She says put it up there on the screen, but
yep.
it's a update.
So basically, recently Jakarta and its surrounding areas have been declared one of the most polluted countries in terms of air quality. The region has really bad air quality, and we believe that one of the most catastrophic greenhouse gas that also comes from human and cattle activity, and also carbon emission, is carbon dioxide. And we think that what if we can take the extra carbon dioxide and convert it into something valuable? And we think carbon dioxide is one of the greenhouse gas that contributes to global warming, and global warming actually increases the prevalence and the distribution of mosquitoes, which can create hazardous tropical diseases such as dengue fever. So we think, what if we can convert carbon dioxide into insect repelling compound? So we seek to assimilate the carbon dioxide from the atmosphere using the XOMP and Calvin cycle, and then convert it into naphthalactone, which is a insect repellent compound. It is originally found in Nepeta, in catnip, if you've heard about it. And so I think it's
Sometimes
not...
you have to drag it onto this screen. No, drag your own. But you may not. We can share your screen.
Yep. Is it second screen only? Oh no, PC screen only. But anyways, this is the putative pathway we're thinking of. So first, because the Pichia itself is an autotrophic yeast already, so it has some fundamental enzymes to be used.
You know it doesn't use CO2.
Yeah, but we can
straight up.
Yep, but we can include some enzymes there, and we're going to incorporate them into the Pichia genome in order.
So Pichia is a bit of an unusual one, just to kind of a very unusual yeast. It's a small number of yeasts of methylotrophs. Okay, so it uses methanol.
Yeah.
And when it uses methanol, so it can use a whole lot of carbon sources, and primarily it does. But when it uses methanol, it turns on a massive amount of peroxisomes proliferate in the cell, and you get the pathway that's fixing methanol through to the central carbon metabolism via DHAP, and you probably got that part of the pathway there. But using CO2 is another matter, and I'm sure you've read the excellent publication of the group from Germany that have been able to engineer Pichia to take in CO2.
So I think we took that paper and combined it with another paper that tries to synthesize naphthalactone. We incorporate some genes over there, 1, 2, 3, 4, 5, 6, 7, 8, in order to convert this glucose into naphthalactone. So that's what we're trying to see here. But then we thought combining these two papers is not really... I mean, it's innovative, but it's not really original. Like, it's just basically taking someone's work, and pasting it. So we're trying to improve this pathway a bit, like probably to push the equilibrium towards the product. So if you have any insights towards this metabolic shunt process.
Well, I do, and I think in some ways, good on you. I think this is a really interesting way of combining these two needs. I think you probably picked, personally, in a practical way, two very low yielding and difficult pathways.
Yes.
So you will have seen from the paper where they've engineered CO2 utilisation from Pichia. It's a very slow process to, you reduce the doubling time of the yeast enormously. And secondarily, these kinds of compounds that naphthalactone is part of are also produced in very low yields. And so you're combining, and along the way, you're asking the yeast to express an enormous number of new enzymes. Okay, look, count them all. Okay, count them all. So it's a whole lot of new protein production just to take in the CO2 and utilize it, and then to produce your nebular-lactone. So as I said, good on you, and it doesn't make it wrong. It just you could get disappearingly small amounts ever made, to optimize.
Yeah, makes sense.
So I think good on you, honourable goal. Personal thoughts would be either emphasise that or emphasise this, and come along with some new ideas about how you might increase yield here or how we might improve this end. And,
so probably instead of emphasizing, like putting an emphasis of both, we will focus and highlight on one part of the.
And bring in some innovation on either side. So either make a much simpler product, or start with something interesting in terms of your carbon source for the Pichia. I'm not dissing your project.
Yeah.
Feel free to go ahead and do it exactly as you like. It's a very grand picture. But you did ask me about practicalities from my point of view.
Yeah.
If I come to practicalities, you'll never make very much of this. In fact, you'll probably need a carbon-13 tracer coming in from CO2 to ever find any of this. Okay.
Yeah. Well, practically this. Another thing that comes out of mind, I mean, because this is technically a design project, so we were trying to make the grandest project ever. But then we were thinking among ourselves, like, let's say if we were actually making this organism, is it actually viable? Is it a viable idea, or will it be just some kind of grand project that will just stutter from the beginning because of this huge complexity regarding all these extra enzymes that should be produced by the organism itself. So we're trying to we talked with another expert from Indonesia, and he kind of suggested that probably we can bypass some of these steps. So instead of, like, let's say, instead of channeling all the carbon sources into glucose, we can channel some of them into, like, let's say, acetyl-CoA, because acetyl-CoA itself is a 3-carbon intermediate that probably somehow can be utilized as a bypass from this huge pathway into the nebular-lactone producing pathway.
And indeed, that's really what usually happens. I wasn't sure why you had the glucose. Most of the time, it goes through DHAP straight into TCA or something like that. You won't find Pichia building glucose from CO2.
Okay, so it's probably something worth to look.
That's a catabolic pathway. Feed it glucose and it will chew it up and turn it into CO2 eventually. This way around, they can already enter numerous parts of central carbon metabolism without going to glucose.
All right.
It still doesn't reduce the number. There's still a lot of engineering needed to make this successful. Okay, and some of that comes down to the very nature of carbon dioxide as well. There's a good reason why. So you have to think, well, where's my electron source going to come from?
Yeah.
CO2 is highly oxidized, and the only things that use it are plants. There's a few other organisms, but let's say plants. They're getting electrons from light to be able to provide reducing power to do something with CO2. You need to think about that. Where are your electrons going to come from?
Okay. To
actually support the introduction of CO2 and its reduction. Okay, because you're going to bring in. So I see you've got methanol here. Is this going to be a co-product, or that can supply your electrons for you for sure?
Yeah. Okay. Makes sense. Yeah. This is something that might not be obvious from the beginning. Like, we need extra electron to in order to make this happen, but that's why we need some kind of insight from you. Yeah.
Have you thought about bioelectrosynthesis, for example? So where you actually supply them with electrons that are renewable.
we might
like literally.
our plan is actually to incorporate all these yeast in a bioreactor that channels like the carbon dioxide from the atmosphere into these yeast, and the yeast can synthesize naphthalene that can be extracted afterwards for manufacturing, distribution, whatever. So I mean, we can actually include some kind of electron synthesizing, electron producing apparatus inside the bioreactor itself. So it might be.
So you know how much of the atmosphere is actually made up of CO2?
Really small.
Yeah, like how much? Like how much?
Less than 1%, pretty sure. Yeah.
So how's that going to magically get into this reactor? How much more are you going to wash over your reactor to get that CO2 to the organisms? I mean, these are practical and they may not be, they may not be things that you're particularly interested in. But let's probe the space, all around, like how you're going to get the CO2 there. Okay. I mean, ideally, we normally want that. If it's going to be your sole carbon source, then you need to offer a concentrated version of the carbon source. It's very hard to deliver very small amounts of very dilute amounts of a substrate to an organism.
Yep.
So that's one thing.
Yeah.
So now we haven't been doing it with yeast, but we've wired up our E. coli and turned it into a receiver of electrons from an electrode, and used it to well, in both directions actually. We've fed particularly rich waste substrates to E. coli. They've generated electricity and products, and we've also turned it around the other way, where we've delivered electrons to the E. coli using a low, highly oxidized substrate. Not quite CO2 yet, because the engineering of E. coli is very hard for CO2, but a low electron-rich substrate, and use the electrons to sort of make up for the loss of electrons in the cell, because you need to maintain a redox balance, and then deliver an enhanced product formation at the other end.
Is there a possibility, let's say, this electron can be, like, instead of creating an electronic apparatus or something to release the electron, like the way you mentioned about supplying electron to bacteria that can regenerate the electron, can we, let's say, use this genetically engineered bacteria and put it alongside these yeast to continuously replenish the amount of electron that they need to push this reaction forward?
Yeah, it's certainly possible. One thing I thought about, though, as I was saying that, the one downside of this is that you have to do that in a fairly oxygen excluded environment, because otherwise oxygen will be your endpoint of electron acceptor. So you will need to then, and I don't know that Pichia copes very well in anoxic situations. So that's possibly off the cards. So
Yeah.
It's just, we need to think about ways. If you're going to use something like CO2, you've got to think about where's the reducing power going to come from, because you won't get any driving force.
Yeah.
CO2 is about as oxidized as it gets. That's why nothing uses it. It's why it accumulates in the atmosphere.
Yeah. Okay.
How are we going to kickstart the metabolism in the cell? Because you'll very soon run out of reducing power. Okay, and CO2 is not going to go anywhere if it's not turned into something that's more reduced than what it is.
All right.
Okay, so that's something for you to think about in terms of your, whether you want to get into practicalities of how you're going to concentrate. Well, how are you going to deliver concentrated CO2?
It's actually good insight. Well, I mean, technically speaking, if we are not supplying carbon dioxide from the atmosphere, we can just use dry ice. I mean, they're basically concentrated carbon dioxide, so we'll just put dry ice there and
It is rather cold.
Well,
it will reduce your temperature.
Yeah.
But this is the sort of thing you need to think about, because you can blast a lot of air through something just to get that 0.05% of carbon dioxide.
So it might not be the best case to channel the atmospheric air into the bioreactors, but probably if we can get our hands on those concentrated carbon dioxide bricks, like the dry ice, we can put it and use it instead.
And there are a number of carbon dioxide capture technologies around, there's no doubt about it. There's the direct air capture, as well as solvent-based ones, there's membrane-based versions, there's all sorts of things. So there are multiple ways that you can get a more concentrated supply of CO2.
Okay. And another thing you mentioned
without
earlier,
freezing the yeast.
Another thing we were talking about is how to incorporate a biosecurity in here. So, because this is a highly engineered organism, we don't want it to be wandering around the environment. So we are going to incorporate a kill switch here, which would take advantage of a different atmosphere, different condition between the bioreactor and outside world. So something we were thinking of, so probably using what was that again?
IPTG?
Not IPTG. We talked about in yesterday's meeting.
Tetracycline?
Yeah, tetracycline. So I think the tetracycline.
I know what the system of the
toxin and the toxin thing. I think using IPTG as an inducer, so we will continuously produce the antitoxin under the influence of IPTG inside the bioreactor, and if let's say spillover happens,
IPTG won't work with PICA.
It won't work with PICA.
Well, it's for a T7 promoter, which is a bacterial one.
Okay, so we have to find out another thing. Yeah, we definitely have to.
It has its own promoters. It's methanol. The AOX1, which is a methanol oxidase promoter, is a very, very strong promoter in PICA.
So
there's a whole bunch of them.
We were actually thinking as well, like, what if, like, we use that AOX2 promoter, and use that as a promoter, and continue to supply methanol, to create an antitoxin. And then once it is out, then there's no methanol, and the toxin will be killing the cells. But I think we scraped that idea because
we could ask yourself first, most genetically engineered organisms are not supposed to get out. Okay, so you have containment measures first, number one, easier to do. Containment measures and SOPs to restrict that. Number two, if it gets out, do you think it's going to have an advantage in the environment?
Probably not, because of the lowest replication cycle.
Yeah, it's like going to be outcompeted by everything that's around. But it will be able to use CO2. But if it doesn't have an electron source, is this thing actually going to survive? And if it did, is it going to be harmful?
Probably not. Yeah. We're just thinking of all possibilities. Like, let's say,
So put in a risk argument. Think about it in terms of a risk argument, and what are the likelihoods, rather than just say, I've got to engineer a solution here. That's you certainly don't want to be adding tetracycline to anything.
Yeah.
Because you certainly don't want to add that out in the environment or whatever. And you're adding actually a problem to the whole system. It might look synthetic biology or whatever, but it's kind of nonsense in this sort of setup, because tetracycline is actually not a very nice chemical. I would argue it on a risk basis myself.
So technically it doesn't really pose a risk, because technically it will be outcompeted by other organism, one, and two, technically there's nothing harmful with all these enzymes, and three.
Yeah, so is the product in itself, would that be the lactone, would that be a problem if it was in the environment?
No, definitely.
Well, one assumes you're actually going to be putting it in the environment anyway, so so it's not making something horrible. Adding tetracycline to it is actually not a very good thing. The fact it could use CO2, but it's not going to go anywhere if it doesn't have an electron source. So it'll probably die.
Okay. Interesting insights, yeah. We were trying to make it really synthetic biology-ish. So we were like, kill switch. And we were trying to make it as fancy as it sounds.
Put a kill switch in there, make it fancy. Obviously put the right promoter on it, but knock yourself out. But in the real world, I can tell you now, you'll be arguing from a mitigation basis and a risk basis.
Yeah. Yeah, because other teams were using all these fancy like RNA structures like aptamer, toe-hold switch or something like that, and we were like, oh my gosh, we were just here inserting a bunch of genes from a bunch of papers together, and we're not going to win if this.
But they were kill switches or whatever, just from the literature. So they're just sticking in something from the literature just because it's
Probably,
an RNA
yeah.
aptamer. It doesn't, something
Okay.
groovy. So think about, you want to win, you want to be innovative. So think differently. Okay, you don't think the same as everybody. Oh, I'll engineer it up. Obviously, you need to have you should have a sophisticated engineering strategy in there. But think differently, okay? What's something that you could do that's different? What could you do, Is there a feedback? How could you get, you could think about how do I get the flux moving here? How do I do that? What kind of different thing could I do by plastering this paper with that paper? How do I keep tapping.
Sorry.
How do I draw this now into this pathway? How do I drive it? What's going to be the way to do it? Can I think about something different to do that?
I think we've come up with some optimization measures. So, for example, so how this nepotalactone works is it binds with the receptor. I wonder if you've heard of TRPA1, a heat receptor or something like that, in insects, and it will probably make them feel hot, so that you go away. So we were thinking, what if we can, from the basis of literature or something like that, we put some kind of modifications to the nepotalactone, like addition of methyl, anything that can improve the binding of nepotalactone to the TRPA1 of the insects, so it has a higher potency of repelling insects away. Another thing that we were actually thinking of expressing the nepotalactone efflux protein on the yeast, so we don't have to lyse them. We were actually frantically searching for the protein for like 2 days straight, and then like during our meeting yesterday, our advisor was just like, probably the reason why there is no paper about nepotalactone efflux protein is because it is not expelled out using a protein. And we saw the chemicals formula, the structure of nepotalactone, and sure enough, it is apparently nonpolar and it's small enough that we think that it might diffuse away from the cell without any protein. I was like, well, there it is.
A little bit hydrophobic, so I'd be wary about that. I would be a little bit wary. Do you have the structure there handy?
No.
It's these, but they're quite right. So small molecules, but when they say small, it's a fair bit smaller than lactose. I think. Oh, okay.
So it's highly non.
Yeah, it's you will get that. You will get that diffusing. Yeah, to at least some degree out of the cell. Yeah. So, and believe me, it takes a lot of energy to break a Pichia cell, and this is a strategy that's being pursued now for people doing, not in Pichia, but in doing, say, Eurovia lipolytica, which is a lipid accumulating cell. People have really liked it for years because you can get huge accumulation of lipid in the cell, and fancy lipids and all that sort of thing.
What cell is that again?
Lipid, fat.
Oh, okay.
Yeah, so Eurovia lipolytica, it's called. I think you can get like 60% of the dry weight being lipid.
Okay.
Triglyceride is triglyceride. But the problem is, you want to get that lipid out, you have to burst the cell, get this unholy mess of cellular proteins and lipid, and you get this emulsion, and then you have to get the lipid out of that. And now they're wanting to generate food lipids within these yeast. We really need new ways of getting to that lipid without bursting the whole cell and having all the contents. So having something that comes out of the cell by diffusion or whatever else is really a great way of going about that. Sometimes if it's a compound that is not tolerated very well within the cell, the organism will find a efflux channel for something, and it will push it out through that. Yeah, so you may find that you get a driving force pushing it out of the cell anyway.
Yeah.
Because some of these things, if they accumulate to a high level within the cell, they
There's
could
a
well.
gradient to diffuse it out.
Well, also you could well have damage, internal damage. It may react with things. It's a reasonably reactive molecule. Okay, so you could have things reacting with mechanisms in the cell, and you'd be surprised at the sort of things that happen inside a cell. You know, suddenly you've got no cell division and all kinds of things like that. So
Yeah.
So, but at any rate, so getting it out of the cell probably doesn't look too hard, and you avoid that lysing and mechanical lysing mostly. You could think about some kind of programmed cell death if you wanted some sort of fancy thing. Get the cells to autolyse, to start a cascade of killing themselves.
After a certain level of metabolic. So, for example, if somehow we can find a promoter that's sensitive to nepetalactone or any metabolite in the process of producing nepetalactone, we can probably use that and then put. Do they undergo apoptosis?
Well, they're eukaryotes, so they have their own. Yeah, they have ATGs and ATGs. Yeah.
We can probably just put like a pro-apoptotic gene down there, and like so whenever there's a combination, promoters in.
And that could double as your kill switch.
Well, it can actually replace the kill switch. Yeah.
And you could have that sensitive to a lactone concentration through maybe put that receptor in there, that receptor from the mosquito or whatever you said, and once you got to a certain level of binding or whatever, start a cascade.
Oh yeah, makes sense as well. Yeah, so we can
because you already know what the receptor is.
Yeah.
So you could say that you're fiddling. You could do some mutation of the receptor so that it becomes a little weaker because of course you don't want it. You could have super high binding ones with a high affinity, and you can have a slightly lesser one, so that you're not killing the cells, and starting a cascade of autolysis from the minute it detects any, and use that as a way to have your internal, level set. I don't know. And you may not feed it back to a cell death. You might feed it back to something else. I don't know. Think about that because you've got a receptor. So do something with the receptor.
Yeah, it would be really cool one if we can somehow like visualize it, like probably try to mutate one or two residues here and there, and it can actually reduce the binding of that receptor to nepetalactone and use that as an inducer of apoptosis. It would be really cool, but well, we only have like 6 weeks left before it's due.
Yeah.
Not sure if it's doable or not, because apart from doing all of this, we are also doing all of these kind of things, interviews, and we have one scheduled at 5. So, but I think some other So these are like the list of optimization we were thinking of. So apart from that modification of nepetalactone, we're also thinking of what if we can just sequester or like scaffold all these nepetalactone producing enzymes in such a way that they are in close proximity to each other and increase the efficiency of the whole reaction.
Works very well. People have done that so we've done quite a bit of work in my group on C1 metabolism using methanol mainly, and definitely in that case bringing those enzymes together in the cascade helps not only to have the product move from one the precursor, the substrate move from one enzyme to the other, but also minimize any toxic intermediates from traveling around into the cell and causing issues. So there's been a number of different mechanisms that you've probably looked at for bringing in a scaffoldless way. And we've also got a technology here where we have a self-assembling peptide that allows proteins to come together, including mixed enzymes and things like that.
Probably not protein, but instead we'll just like bind the genes together in like a single plasmid, so it's located closer to each other technically. Well,
that doesn't stop
it does,
going all over the place when they're expressed.
Yeah, but we're still piecing together the pieces of the puzzle. But yeah, I mean, if we can somehow come up with a protein structure that can be docked by other proteins in the vicinity, it can actually. And
that's another. So we have a technology that we've developed here too, where we anchor proteins, enzymes, all sorts of proteins, into the lipid droplet, which is a lipid-filled organelle. It's a budding off of the ER, and the proteins are displayed outwardly into the cytosol, and that's another way of keeping them together. And it also means you can isolate them too and use them if you want to. But with a small protein that docks into very specifically into the lipid droplet, you can then have this nice scaffolding and nice way of keeping the pathway together.
Okay.
And that's just an extra bit of protein on each one of those genes, so like a bit of gene on those genes, and it's expressed as a fusion. So you don't have to have partners finding each other in the cytosol and then coming together and going in there.
So it's already scaffolded there. It's easier.
Yeah, and
that's what we are thinking, but it's
very easy to do.
Okay. I would say it's easy, but
Oh no, I'm just saying it's very easy, and you're not actually going to do it. But so we've demonstrated that you can do it on the N on C terminus, depending on which way you want to do it. Works with all signaling proteins, works with enzymes, works with all sorts of things.
So how would you do this? Like, how would you design this linking thing, like so that the enzymes are related to each other?
Yeah. So as you said, big plasmid or whatever you have. If it's a big plasmid or a big insert into the genome, each one of so it can be you could put it in as a long read, but remembering that each protein that you're putting in, each enzyme, needs to have, it needs, a promoter or a initiator of some sort, and then, so we have this, what we call oleosin, which is what we don't call oleosin, the protein, the short protein that docks into the lipid droplets called oleosin. That's just expressed in one fusion with your gene of interest. So one of your enzymes plus oleosin. Another one of your enzymes plus oleosin. Another one of your enzymes plus oleosin. And then it gets expressed as you might integrate it or have it as a plasmid. And then each one of them gets expressed, and they will all dock into the lipid droplet.
Wow, that's actually interesting. I think I read some of the papers posters up there. I think I vaguely remember oleosin being mentioned somewhere.
Yeah, there's one out there.
Yeah.
And the other one we have is a peptide that, when it finds a peptide of a similar type, under certain conditions, it will come together. They will spontaneously come together in a particle.
Okay.
That might cause all sorts of things inside a cell. They don't like things assembling that look like viruses or whatever. So I don't know what would happen inside a cell if you try to assemble in there. But yeah, that's another way of bringing them together. But yes, it's very important when you've got a pathway to try and co-locate the enzymes so that you don't have intermediates drifting off. It just reduces your conversion enormously.
All right. And when you're trying to make like these kind of metabolic engineering, of course you have to somehow perform mathematical modeling to test this. Like, do you have any resources that we can use? Because honestly, all 8 of us has no prior experience regarding like mathematical modeling. I mean, we're biologists after all, so mathematics is not our best friend. but do you have any particular resources that we can trace
There are publicly available models for good ones, for certainly excellent ones for Saccharomyces, which have been updated over the years.
Sorry, what was that again?
Well, they're genome models, so they're based on what's known to be expressed pretty much in these cells. So there's definitely ones for P as well. Now, you do need to have some understanding, because otherwise it's garbage in, garbage out, and garbage out. But these models are publicly available, and they can be just based on the existence of these proteins in the cell, or they can be flux based. There's a number of models that actually have measured fluxes, like for example in your metabolic pathway you were showing before, in Pichia, what's likely to be the flux from acetyl CoA into the next step, or into the tricarboxylic acid cycle or whatever. So some of these are flux-based models, because people have done carbon-13 tracer studies where they might have fed it glucose or whatever, and they know what the fluxes are for the main routes within the cell. Now, I don't think you can go and get a genome model yourself and have a play, but it does require a little bit of computational knowledge. We don't do computational modelling. We collaborate with people who are expert in that area.
mathematical modeling?
Yeah, so they don't build the models themselves. They actually they take these general models off that are publicly available. They may manipulate some of them because they might add in a new gene or something like that, and then what they're looking for is, okay, what's the ramification of that, or how can I improve the flux into my product down here, or might be trying to rationalise a set of data. You know, they've tried to put in a pathway for nepentolactone, and then they find that at the other end, you know, they're only getting precursor here, getting almost nothing, almost nothing. And why is it getting stuck there? You know, so
and that's when you pinpoint a problem and then try to rectify it.
That's right. So it's requiring a precursor from the tricarboxylic acid cycle, which is at very low flux in PIHIA. So an aim for your metabolic engineering will be, okay, we need to improve the flux here so we can get more precursor there, so we can get more down here. Yeah.
And apart from like mathematical modeling, I believe that there are like some tools that have been developed for metabolic modeling as well, like the ones that has these large metabolic pathways of the cells, and then when you try to alter it, you can visualize what will happen afterwards. So do you have any suggestions on what kind of tools we can use for this?
Yeah, so you mean for yourselves as opposed to research that may be done and then people. So people do metabolomics, and they may well have done metabolomics on nepentolactone synthesis within certain organisms. They will have pathways, and they will understand where there have been choke points, or where certain model interventions have increased flux or whatever. So you'll see, kind of diagrams with big arrows and things like that, or all the metabolites are stuck up here and nothing down here and that kind of thing. I don't know. There's some maybe that are a bit dynamic. I'm trying to think if there's any dynamic models. That's what you're talking about. If I do this, will I get more there and that kind of thing? Is that what you're talking about? Certainly not we use. Yeah. And you need to be aware that a lot of the models that are out there are for unengineered organisms. So you put your pathway in, it doesn't immediately tell you what's going to happen to your pathway. You then have to extend that model. You take that generalized model, you put in pathways with some parameters, KM, some maybe some kinetic parameters for the pathway, and then you try and meld those to the existing generalized model and your new pathway. So, I'm not one that you can just do yourself.
It's gonna be hard.
Well, you've got other tools. So if, for example, you go and do the engineering of your receptor, you could, you've got all the, you can visualize 3D.
AlphaFold.
Yeah, and you got, PDB. You can go in there and have a good hard look at it, and make some changes and make some guesses, make some hypotheses about what's going to happen. There are binding. There are software for looking at docking. Very easy, that one to do. So what kind of modelling? Nobody's mathematically or computationally gifted. I'm trying to think of the sorts of things that we look at routinely. And you'd be aware of all the sort of normal bioinformatic tools, wouldn't you? Just whole suites of things that are around, the normal sort of things, KEGG and all that kind of stuff.
What was that again?
KEGG.
I think it's like a database of something. Right?
Metabolic pathways.
Yeah, KEGG.
Really, really detailed. There's so you haven't looked at some of those sort of more openly available metabolic pathways.
Not because we were focusing on creating the genetic circuit first at this point. So our Gantt chart right now in October, it's time for us to optimize and to perform mathematical and metabolic modeling, and then optimization, and then in November we're going to finish the whole report, create the presentation, the posters, and the stuff that are required for the competition. So
So you might need to. Do you know what kind of level of modelling is expected? Because I think you're meant to, normally in these iGEM teams and things, you're meant to have some sort of model on your team, aren't you?
Yep, but well, technically we were searching for one. We get a recommendation one from a Canadian university. He's a really good computer programmer and also has some solid robust math background, but he decided not to join because of his other commitments. But we can probably try to seek for assistance. Well,
I've got a PhD student who's just finished, who he's back in Indonesia at the moment, actually on holidays.
okay.
Because he's been in Eng, he's quite mathematically, and he's well, he's at least more accustomed to it than probably the rest of your team.
Yeah.
Now, whether he'd be interested or not, I don't really know,
Yeah.
but be happy to put you in contact. He doesn't do modelling per se, but I'm just saying he's much more, kind of mathematically aware and things. So,
Is it possible to put us in contact?
yeah, be very happy to.
Yeah.
I mean, all he can say is, I'm
Great.
not interested. and and I can sort of point him in the direction of some of the tools. But
Yep.
we've had discussions before about him potentially moving into a more bioinformatic area. And he's expressed a bit of an interest, but he's also thinking about starting companies and things like that too. So he's right at that stage of really making a decision about what he wants to do next.
Yeah.
And this is fairly short term that you're talking about. So.
It is really short term. But I mean, it's not impossible that if this project goes on, we might actually try to make it happen. Well, it's just that this is more of a designer than a practicality thing. It's not like the actual iGEM competition. Like how the actual iGEM competition is really hands-on project and it's really expensive, tiring. But this is just an online in silico.
I think it's a great idea to do it. I think it's a really good thing. I mean, it stimulates your thinking without having to, without, as you say, you don't have the wet experimental requirements. And I think it's a great idea. Frankly, iGEM's not available for everyone and not available, for all sorts of reasons. But this is a great way of having people still, mentally teased. So I wouldn't be concerned at all. I think it's great because sometimes if you're just thinking about all of that. Okay. I see you got the grow L and grow S there for bacterial chaperone.
So I think the grow L and grow S were chaperones inside the peroxisome for the pathway, for the XOMP calcium pathway. Our advisors were thinking of, instead of using those chaperones, can we use another chaperone for bacteria instead of these two? Because, well, inserting one gene is better than inserting two genes. Or probably choice of promoter, we're trying to play around with the promoters, like is there any kind of way to improve the yield by changing the promoters. We've actually come up with a rough design on how the path of the circuit will actually see. So like, this is the rough design of the CVP cycle. We haven't made
Rubisco. You put Rubisco in.
Well,
you have to for your CO2.
And then we haven't made
it's
it
massive.
Well, it is massive.
Yeah.
But then we haven't made the schematics for the biosynthesis. I think we were meant to make the whole thing today. And the coil switches. That's what we were thinking of IPTG, but we're gonna scrap that.
Well, you have to put a T7. You'd have to have a T7 promoter in there if you're going to. And I don't know. It won't probably be recognised by the machinery in PICIA. So.
Yep, that's something that has to.
And also the what was I going to think of? So what you need to do is match your chaperone with where the protein originally came from.
Okay.
Okay. So the nepetalactone's from a plant. So you'd be much better off with the plant.
For the Nepeta chaperone.
If it's anything known about whether there's specific chaperones to help that pathway fold, the enzymes in that pathway. Is there something unusual about the enzymes in that pathway that they might need a chaperone?
Honestly, not sure. But we'll take a look at it. Yeah.
And so that's But anyway, I'd be happy to shoot Kevin an email and see whether he's interested in,
really
also sort of suggest to him, because as I said, it's not work that he's done before, so he doesn't. It's not like he, gem model of pick here, and I'll just put that in. No, it's way different from the sort of work he did in his PhD.
Okay.
So I was an associate supervisor, so it's not really. He wasn't really in my group. So
it would be great. I mean, if he's interested to help us, it would be amazing. What other questions do we have? Let me pull up the questions we've formulated for you. No. Next question is, like, you're focusing in Saccharomyces cerevisiae, but like, what are the main differences apart from peach has autotrophic yeast? Like, what are the main differences between these
it's a heterotroph.
organisms? It is.
It's definitely a heterotroph.
But we can. Okay. Hetero. Yeah. Okay. Hetero. Okay.
Bunch of engineered it.
We are engineering.
Yeah,
Yeah.
the point being, you want to engineer it to be an autotroph.
Yeah.
Autotroph. So, what's the main differences? Well, so it's Crabtree negative, so it doesn't make ethanol. Whereas Saccharomyces, you give it to a lot of sugar, it will immediately make ethanol.
Okay.
Pichia never does that, and so you can get higher cell densities when you're growing it, and you, in some ways, but also it's an obligate aerobe. Saccharomyces can work in anaerobic as well as aerobic conditions. So there's pluses and minuses to that.
Yeah.
They, in terms of protein secretion, Pichia is supposedly superior, and certainly in our experience it is. Mainly because you can get really high cell densities. You get high cell densities, you can make a lot of protein.
Makes sense. The other questions are done. Do you have some suggestions or some words for the synthetic biology community in Indonesia, based on your experience? Do you have any wise words or some suggestions?
Well, I think it's certainly because it's a technology that can help all countries. Doesn't always have to be only. Well, it's not. Might have started in more developed, more Western countries or something, but it really it's a technology that's got a lot to offer everybody, and whether that be low cost ways of making lactone or vaccines, particularly, which is something that's really high on my list. It can actually be a way of redressing some of the balance where in some parts of the West where some things are very exclusive and very expensive and therefore only available to a few people in the world. Particularly if you're talking about microbes, you can do things cheaply. People have proved it for many, many years. You can do things cheaply. You can make ethanol really cheaply. You can make a whole lot of products very cheaply using microbes. And so it doesn't have to be only a technology that's very, very hard to do and very, very sophisticated. It can be, but it doesn't have to be. And I think more people investing in it, particularly countries with a lot of natural resources to be able to turn carbon-based products into things of high value, then it's a perfect technology for that. There are plenty of countries that, I think Indonesia does have a reasonable amount of oil, but there are plenty of countries that got no fossil carbon at all, no coal, no oil, no nothing. But they might grow a lot of biomass. That's a resource. So that's probably the only wise words. That's the kinds of wise words. And it's great to see, and be excellent to see a whole Southeast Asian build up of synthetic biology and and its application.
All right. One last question.
Mm-hmm.
If you can be a gene, what gene would you like to be?
Oh, if I can be a gene. Oh, dear. I only get one gene.
Well, you can name as many as you like.
Because if I was going to be one gene, I'd want it. Well, then you can't be part of a really big complex. Well, I think I'd then have to choose. Say, there'd be an obvious one. There's this protein I've just worked with pretty much for many, many, many years, and it's everywhere. And that makes it sort of innocuous, but at the same time makes it super important. So it's a thing. It's simple. It's a simple protein. It's a simple enzyme. It's called carbonic anhydrase.
Okay.
It does a simple job, turns carbon dioxide into bicarbonate and a proton. But it keeps all life alive because it allows us to buffer and remove CO2 in capture it, to eliminate it, or to do something with it. It's so versatile. It provides protons for our digestion. It removes CO2 from our respiration. Or everything has it, from the simplest bacteria to the most complex organism. Would be a carbonic anhydrase.
Simple, elegant, yet super important.
That's right.
That's your motto. All right, I think that's all. Thank you very much for your time.
Pleasure.
We have a little token of